is Dr. Saraswathi Madam, Principal Scientist, Horticulture, ICAR-NRCB, Trichy, to deliver a lecture on advanced micropropagation techniques. Madam has completed BSc Horticulture, MSc Horticulture, and PhD Horticulture. And she worked as research associate, scientist on senior scale, senior scientist, and presently working as principal scientist at NRCB Banana, Trichy, since March 2014. And for her significant accomplishments, Madam has received several national and international awards. And among them, she has received Best Oral Presentation Award as well as Best Poster Award during 2020 at International Conference on Innovations in Sustainable Production and Value Chain Management in Banana, organized at NRCB Trichy. And Best Poster Award during 2016 at First International Agro-Biodiversity Congress held at New Delhi, and NAIP Overseas Fellowship ICAR during 2011. And she has also received Best Poster Award during 2010 at Global Conference on Banana held at Trichy. Then she is a working group member of Biodiversity International France since 2003. And she has also received Best Poster Award during 2002 at Global Conference on Banana and Plantain held at Bangalore. And Madam has also received Chai Best Paper Award during 2019 and Fellow of Horticulture Society of India during 2018, Fellow Chai Award during 2018, Best Presentation Award at IIHR during 2016, Best PhD Dissertation Award at AIPUB 2010, and Thiru T. Gopalan Nair Memorial Medal at TNAU Coimbatore during 1994, and Indo-American Hybrid Seeds Medal TNAU Coimbatore during 1994. And she has also published several papers, research papers of 46 number and 21 popular articles, one book, 10 book chapters, 10 technical bulletins. Then she has also presented 15 papers at various national and international seminars, conferences, etc. And also presented posters at all those seminars and conferences, and published 115 abstracts. And to her credit, she has also guided 6 PhD students and 30 MSc students, and published several papers. And there are so many projects in which Madam was involved as PI or co-PI. She has ongoing research projects are framing crop-specific dust guidelines for banana, and development of trait-specific markers for Fusarium wilt resistance through association, mapping studies in banana, and improvement of grandini for Fusarium wilt resistance through non-conventional breeding. And she has also completed 6 projects on banana, which were sponsored by DBT. So this is the brief introduction about Dr. Saraswathi Madam. Now I request Madam to please deliver a lecture on advanced micropropagation techniques. Thank you, Madam.
Thank you so much, ma'am, but it is a very long introduction. Thank you. Shall I share my screen, ma'am?
Yes, madam. Yes, madam. I have given permission. You can share it.
Yeah. My screen is visible, ma'am?
Yes, madam. Madam, can you keep it in slideshow mode?
Slideshow only I have kept.
Here it is not in slideshow mode, madam.
It's not in slideshow, ma'am.
No, madam.
Now,
Still, it is looking like that only, madam. It is not in
I
slideshow.
don't know, but I have put it only in slideshow. Shall I continue, or maybe if there's any issue?
Okay, madam. It is visible. It is
It is
clear
visible,
only.
right?
Yes. Students, please keep your mic in mute.
Okay, shall I go ahead, ma'am?
Yes, madam. Yes, madam.
A very good evening to all of you, especially to Madam and Sir who have joined this meeting. And dear students, I hope that this lecture will be very much useful as you people are getting ready for your graduation. Okay. I'll be talking about the basic micropropagation as well as the advanced micropropagation techniques and their potential applications in banana. And I think I need not tell all these things. You people know that in the present day plant tissue culture, it has a major thrust area in biotechnology, and this where we are making use of the totipotency, that is the capacity or the potential of any tissue, cell, or organ when it is grown under a synthetic or an artificial medium under controlled condition, it is able to regenerate into a full plantlet. That is the totipotency. And if you just look into this plant tissue culture, which is a branch of biotechnology, it plays a vital role in second green revolution because in the sense where we are working on genetic transformation and other marker assisted breeding and other aspects of biotechnology to improve the yield and quality. So if you see, like this forms a basic in vitro system for genetic transformation as well as for the mutation breeding studies. And a differentiation, that is what is happening when it gets converted from any cell tissue or organ into a plant. And de-differentiation, that is when from that organ, if we are trying to develop the callus, that becomes the de-differentiation. And re-differentiation is one where again from the callus we try to develop the plantlet. And as you all know that banana, it is the world's most exported fruit, and it is the fourth important crop after rice, wheat, and maize. And this is a crop of small-scale farmers, both in the developing countries of the tropics and subtropics as well. So it becomes very essential to increase the production and productivity of the crop, despite the challenges that that is faced by the banana cultivators or the farmers to say. So if you look into the major pest, I think you all are aware of this, but as a prerequisite, I'll just fastly just go through the slides. So the rhizome borer or the corn beetle, it is an important pest, and pseudostem borer you people know, and scarring beetle, which is a major problem in North India. Everywhere you can see that the fruits, particularly it is full of scars like this, which actually spoils the
Excuse me, madam.
yeah.
Madam, slides are not moving. Only that first slide, advanced micropropagation techniques and their potential application slide is only on the screen.
Oh, but I have put the next slide.
Uh,
Now you
not moving. No, madam, that first slide only on the screen now we are seeing.
now, sir. Now,
No, madam, not moving.
oh, I don't know. 1 minute, sir.
Madam, slides,
Yeah, I have kept it in the, anyhow, let me stop share and I'll do it. Sir, now? Sir?
Now, madam, this is just slide is in now.
Now this is best slide?
Yeah, best.
Okay. Sir, if
You
you
just-
don't visualize the slides, you just tell me, sir, because here nothing goes wrong. It's okay.
Okay,
Yeah.
nothing. Okay, madam. Now we are seeing that
Okay.
pest side.
Yeah. This you can see the fruit scarring beetle damage where it affects the aesthetic value of the fruits. Though the quality of the fruits not much change, but it affects the aesthetic value. And even if we see in the market, we will not have the we will not get attracted and we will not definitely buy these type of fruits. But if you like, I have traveled in many parts of North India and I have seen that all fruits which are in the market also it is fully damaged by the fruit scarring beetle. And aphids and thrips, it serves as a vector for the banana viruses and nematodes. You can see the major three nematodes are the burrowing nematode, root knot, and the root lesion nematode. Sir, it is second slide. Is it moved, sir?
Yes, madam. Yes, madam. This is yes.
Mm. And you all know that the major fungal diseases are the leaf spot and the fusarium wilt. And if you look into the bacterial diseases, that is the Ervinial rot, which is now Pectinobacterium Ervinial rot, which is a quite common problem in case of tissue culture plantlets, because the tissue culture plantlets, they are highly susceptible to this Ervinial rot. And viral diseases, these are the four major viruses, that is the banana bunchy top, black mosaic virus, streak virus, and the cucumber mosaic virus. So these are the major viruses which are present in the banana. Like despite all these changes, challenges, like if you see in the past 1 decade, we are able to increase the production and productivity. This is mainly because of the adoption of the improved production technologies by the farmers. Like they go for high density planting, they go for fertigation, and they go for integrated pest and disease management. So all these have contributed for the increased production. And the most important factor that has contributed is the quality planting material, the use of quality planting material, and that is possible only in case of tissue culture multiplication. And as such, maybe these are all the basic slides, but still, I just want to re-emphasize on the fact. And you know that through micropropagation, you are able to multiply rapidly. That is within a span of 10 months period, you will be able to produce almost like a from one sucker, and you will be able to produce almost 1,000 plants, which is the maximum as in case of granite. And minimum, if you see, in case of all the traditional banana varieties like your Amrit Pani or Rastali, whatever it is, the number of plants which are produced per sucker, they are very less, and it ranges from 250 to 400 numbers. And what is called the product uniformity, because on the tissue culture plant, they are produced as they are produced from a single mother plant. All of them will be of uniform age. So when you put them in the field, all of them will come to uniform harvest. That there will be normally there will be a staggered harvest, and it will run for 1.5 to 2 months. But in case of tissue culture plants, it will be, you'll be able to complete the harvest within a span of one month, which is again advantageous. Thereby, we are able to save the labor and cost, and also the management becomes easier. And disease-free planting material. This is quite obvious because we index, like whatever the mother plants itself, we see that it is pest and disease free. visually, and we also index them for viruses. So all these aspects are taken care of to result in the production of disease-free planting material. And plant movement, in the sense, like if you transport the suckers in one load or lorry load, maybe for example, if you are able to transport 1,000 suckers, the same in case of tissue culture plants, you'll be able to transport almost 10,000 plants. That is, it makes the movement of plants from place to place easier and higher returns. Though these plants are highly tender and they are like test tube babies, but if you take utmost care and if you provide the utmost care and proper irrigation and proper nutrition, everything, then it is likely to yield 10 to 15% higher when compared to the conventional suckers what is normally being used. So that it ensures high returns to the farmer. And season independent production, because under in vitro condition or under controlled condition, you will be able to produce the plants all through the year. And this is you can see the uniformity of the product which I was talking earlier. This is in a tissue culture plantation at Deni, and the factors involved in the tissue culture multiplication. And this identification of elite mother clones, it is most essential, because some of the tissue culture facilities, what they have their own mother nursery, in the sense where they try to grow the elite plants under screen house conditions so that it is not affected by major pest and diseases, and it is highly ensured free of pest and disease, and you can collect the explants from them. But most of the tissue culture companies, they don't have their own mother nursery. So what they do is they take up a survey across the farmers' fields, and they tag the plants, so that they will collect the explants only after harvesting, or they will go tag the plants once they have put forth the bunch, and they will wait, and once after harvest only they will collect the explants or the mother plants, we can say. So they don't stop with that. Once after coming to the lab, they will index them for viruses, and also they will test for the clonal fidelity. That is because you have several ecotypes, of maybe your Rastali or Amrit Pani. So they will test for the fidelity, and they will ensure what they are multiplying is, like for example in Cavendish, you have several types. You have Grand Name variety, you have Shrimanti, you have Madhukar, and you have Dwarf Cavendish, you have Robusta type. So many types are there. So if the company aims to produce plants of Grand Name, they have to ensure that it is only Grand Name, and they don't do a multiplication of varietal admixture, because that cannot be accepted by the farmers, as it might lead to loss because of non-uniformity in flowering, and the bunches may not be of the same quality. So all these factors has to be ensured, and that is possible only by testing for their clonal fidelity. And mother clone nursery, as I told earlier, and aseptic culture initiation of index plants. Definitely it has to be aseptic culture. But as far as banana is concerned, you have the problem of endophytic bacterial contamination, which happens, which is expressed even at the stage of shoot proliferation. So all these cases, you have to ensure that you don't end up in contamination, and you are successful in the establishment and multiplication of plants. Then the multiplication, after multiplication, what happens is, first will be the initial establishment, next will be the shoot proliferation, third will be rooting, and then comes the hardening. So, and the companies, they double check or double ensure that because they test for viruses and clonal fidelity at different stages. Especially now, it is being insisted by the DBT NCS TCP that they have to index the mother plants as well as the secondary hardened plants. And the clonal fidelity testing, it is being done only at the hardening secondary or the hexagar plants only. We test for the clonal fidelity. So this is how we double ensure that only quality planting material is being supplied to the farmers. And identification and regular roguing, this is most essential. You people know that somoclonal variation is a problem. So even in the nursery, they try to look for the off types and they try to cull them out so that the farmer doesn't face any problem in cultivation of tissue culture plants. My slides are moving, sir. Sir?
Yes, ma'am, moving.
Yes. Ascertain the trueness of the mother plant. That is what I was talking about, the clonal we always like I'm not sir, you are not audible for me, so let me just go ahead.
This is visible. Slides are moving, madam.
Okay. Okay, sir. Like we should ensure that at the time, like they should be healthy and they should exhibit a normal growth. And as I told earlier, we should always, when you tag the mother plants,
Go ahead, madam. Slides are moving, no problem. Please go ahead.
okay, like we should ensure that it is not only free from pests and diseases, but also it should have a high yield because that is the interest of the farmers. So at any cost, they shouldn't lose their yield. So that is why we always uh, insist that the mother plants or the explant should be collected only after it comes to bunching stage, so that we check whether it is true to type, where like visually, not by markers or molecular markers, we just check visually, and if the fruit characteristics are similar to the grand name, and then only we collect the mother plants. And visual freeness also we have to check. And this is some possible physiological disorders. You people would have known that fruit skin splitting, it is a common problem in all Cavendish clones sometimes, and also in Marburg and Napoleon. Marburg is nothing but your like Amrit Pani type, which belongs to the silk group. And hard limbs also, again, it is a problem in case of Amrit Pani silk type. And unusual soreness, that is, you can see that the acidity is more when compared to the sweetness, and acidity is the dominating taste of what we get in some clones of Puvan. So such types should be avoided. And seediness, again, it's a problem in case of Puvan and Karpuravalli, where you will have enlarged ovules. And soft tissues, again, it's a problem in case of Cavendish and red banana. And unusual ease of fruit detachment upon ripening, so that when you just pull out, it comes out easily. And that is also, it's not a favorable trait or a quality trait. So, sir, is it visible?
Yes,
Sir?
madam. Yes.
Yes. Yes. Basic techniques, like you know that preparation of culture media, that is quite that is the basic step. And sterilization, you have to sterilize both your whatever the glasswares, whatever the minor equipments what you use, and also the media you need to sterilize. So all these comes under sterilization process. And explant preparation. So explant preparation, again, it is a quite important in the sense of what should be the size of the explant that we put it in the media. That also plays a major role, because some companies, they put the shoe tips. Maybe when I show the subsequent slides, you can see how small the shoe tip is. And some of them, they will put a larger shoe tip, because when you as per my experience, when we put the larger explant or the larger shoot meristem, what happens is the surface area is more, and naturally the phenolic exudation will be more, which will inhibit the further growth. Not in all cases, in some cases. And next, what we do is inoculation. And inoculation, again, you have to do it under laminar. And incubation, definitely you have to incubate them in the culture room with all the controlled temperature and humidity. And acclimatization is the hardening phase. And these are the sterilization methods, that is physical sterilization, where you go for autoclaving for glasswares, and hot air oven for again for physical sterilization of glasswares, and even for drying we can use, and UV radiation. Because normally when just before we start the work, what we do is whatever we are going to use, like your glasswares and your forceps, scalpels and the media and the petri plates and whatever the culture plate, everything you need to, we will put it in the laminar airflow, switch on the UV for 15-20 minutes. Then only we will start the work. And chemical sterilization, you know, normally ethyl alcohol, ethanol we use it, and mercury chloride we use, and sodium hypochlorite, calcium hypochlorite, all of these are some of the sterilants what is being used. As far as the banana is concerned, we use ethanol when we go for the male flower bud culture, and the mercury chloride and the sodium hypochlorite combination when we go for
Ma'am, Saraswati ma'am, just now again sites are not moving that selection criteria for mother plants only seen on plants, that side only on the screen now.
again
Madam?
slides are not moving, sir.
Yeah.
Yeah,
Now,
1 minute.
selection criteria for mother plants, that slide is on the screen now.
Oh.
Some problem.
1 minute. I will stop sharing and then share it again, sir.
Okay.
Yeah. Sir, is it visible?
Yes, madam.
Yes, sir. I don't know what is happening. But
Sterilization methods. It is
yeah,
now sterilization methods. It is
yeah,
there.
yeah, yeah. And filtered sterilization, you people know that most of the growth regulators, like a non-BAP and other things, like some of the new generation growth regulators, like your thidiazuron or metatopolin, they all of them they are thermolabile, and you cannot add it to the medium and then sterilize. So you need to prepare the medium, autoclave, and then one little bit you have to cool. Then you have to add this growth regulators using the microfilters. So that is what filter sterilization. And now let me come to the subject, that is shoot tip culture. Normally we recommend that you have to take 2 to 3 months old suckers, and from that swat suckers. Okay, it's not a water sucker. You have to select a swat sucker, and from that you have to extract the meristem, which is of 3 centimeter cube, and then you have to put it for the in the culture medium. Normally the subculture cycles, normally what we recommend is that it should not exceed 8 subcultures, so that because when it exceeds 8 subcultures, then studies are there that they result in more percentage of somoclonal variants, which is not desirable to the farmers. Then again, that's what. Now this is a this is a tissue culture lab, which is a modest facility only what we have, and maybe now we are planning to have a commercial tissue culture unit. So this is a optimal one, and you can see this is a SWOT sucker. And my slides are moving, sir.
Yes, ma'am.
Yes.
Yeah. This is a SWOT sucker what I was talking about, and this is a 3 centimeter cube meristem what we normally extract, and that we establish, and once after establishment we induce the multiple shoots using BAP or IAA, that is BAP IAA combination, and that again that varies with the genome and the variety what we use it for multiplication. And normally while in single plantlets are produced, there will be roots attached to it, but those roots they are not quite active. So what normally we do, we put it in exclusive rooting media where we supplement the media with either with IAA or IBA or NAA. Then we again reproduce the roots, and then only we go for hardening. So now these stages, like earlier we were doing one primary hardening, which will be done in pro trays, and then we will go for a secondary hardening in polybags where we use soil mixture, and after the completion of the secondary hardening it goes to the feed. But now like just to reduce the hardening period, because normally this is 15 to 20 days primary hardening, and the secondary hardening, it takes normally 1 month, so totally 45 days. So now to reduce the duration of the hardening phase, what is being done is compressed cocoa peats are available, which are available in the tablet form, which we can put it in water, so it swells up, and in the same thing we can do both the primary and the secondary hardening, and we can little bit reduce the hardening period by some 10 to 15 days. So that is the advancement what we have done. And this is like a meristem block culture. This has not been so much successful, but I have tried it on my own. But and this I learned in a Taiwan laboratory where I went for a training on tissue culture. This is what this is normally the explant what we put it in the culture initiation. So, but in this case, we try to remove all the whorls and we try to remove this meristem as a block. You can see here, we just remove it as a block and we put it in the medium. So what happens here is normally our multi-shoot or multiple shoot proliferation, it happens in the third or the fourth subculture. But when we use the right media and as well as the right technique of handling this block tissue, that is meristem block, you will be able to induce the multiple shoots in the very first subculture. So that is the advantage of this method. And all this time I was talking about the shoot tip culture. Now this is like a male flower bud culture. So you know, this is the male flower bud. So after the opening of the last hand, we wait for 1 month, and immediately after that we try to take out these flower buds and we try to put them in the, we remove bract by bract, and then we remove the florets, male floral hands we can call it as, so that we put it in the medium. So after that what happens is, the floral meristem, it gets converted into shoot meristem and it forms a meristematic clump. Then after that it follows the routine procedure of multiple shoot proliferation, then rooting, it comes to hardening. So what is the advantages of this method? Here, as in case of shoot tip, we need not go for a long sterilization procedure. That is explant sterilization I am talking about, because here just we remove all the bracts and we bring the size. Maybe in a subsequent slides I'll show the size of the male flower bud which we are taking for culture initiation. So that will be hardly 5 centimeter length, because we will remove all the outer parts and innermost part will be only 5 centimeter size. We will take it to the laminar airflow chamber. We don't do any surface sterilization. We just swab it with 70% ethanol, and then we remove the minute floral bracts with the help of the scalpel. We cut or we exercise the floral hands, and then we put it in the medium. So here, this, because in the subsequent slides I'll be talking about the somatic embryogenesis in banana, because all the floral whorls, which is from like you make your count from the outermost whorl. So if you take all the bigger floral hands, florets, we can call, these are all the florets. So all these will, we can, if you use the appropriate media with the appropriate growth regulator combination, you can go for a direct regeneration protocol as it is mentioned here. And so totally it is free of contamination, because since the flower buds innermost part, what we take, we just swab and then we take it to the laminar airflow, we do the culturing. So it is totally free from contamination. And you can see from one flower bud of 5 centimeter length size, we can produce 1.5 times more. That is in case of, you take in case of a granine. If you are able to produce in case of sucker, that is shoot tip, if you are able to produce 500 plants, using this one from single flower bud, you'll be able to produce almost 750 plants. So that is the advantage. And this is a direct regeneration from comlet explants. So this is we do when we have hardly one sucker, and which we are not sure enough that whether we will be able to establish them under in vitro conditions. So in that case, what we do is we just bring the suckers, give all the treatments, necessary treatments like fungicidal treatment, we shade dry, and then we use a sterile pot mixture. That is, normally we use sawdust. We use it, and that is a sterile one. We put the sucker, whatever the one sucker of a precious clone or a elite clone, whatever we have collected, we put here, and then we try to give the break the apical dominance as we do it under in vitro conditions. Because once we break the apical dominance, what happens is all the multiple shoots, all the axillary buds or the lateral buds, all of them, they get activated and they start growing up. So this is how it happens. And now each plant will have a mini sucker, which we call it as the comlet. So that case, you'll be able to rescue that one particular precious material what you have. And here also the multiplication rate is faster, that is 1.5 times more. So this is the advantage of this method. And let me come to the low cost tissue culture protocol. And normally all these factors like chemicals, the labor, the energy in the form of electricity, and the capital, it contributes to the production cost of the TC plants. And in that, if you take the chemicals, the major expensive materials are the agar, the gelling agent, and the carbon source, which is the sucrose. That alone contributes to 60% of the chemical cost. And if you take chemicals and energy and electricity, it almost contributes to 40% of the production cost. So these are the areas where we can intervene and we can try to cut down the production cost. Because like normally, these are all quite a bit old concepts, but still they have the potential. Like if you take the tissue culture laboratories, none of them they are using double distilled water. Now everybody uses only RO water as a for media preparation and for all other aspects of tissue culture. Like they just autoclave the like RO water only, they just use it, and for other sterilization, everything they use only RO water. And chemicals, if you take gelling agent, earlier days we will be confident only if we use agar. Like or even more expensive than agar is the phytogel or the clarigel, which is more expensive. So now we try to use even the low cost gelling agents like even sago you can use, or your plant ovata, that is your isabgal seeds. I hope most of you would have seen, when you put them in water, it absorbs and it forms a gel like substance. So exclusively we cannot use them, but we can use 60% of this low cost component along with agar. And similarly, sucrose nobody uses now; they use only the market sugar, whatever is available. So that we made a study and we came to the conclusion that they can be used without any problem in the multiplication of the plants and even in the field establishment, or there was no change in the clonal fidelity of the plants which were produced out of the low cost tissue culture media. And further, all stages could be accomplished, like initiation you can do, and this is a multiplication you can do, and even the rooting phase you can establish. So if you take this is now the cost would have enhanced to 70 rupees maybe. The per liter cost, if you calculate for media, when we use the high media compounds, it costs around 60 rupees. So, but when we use try to substitute them with the low cost components, then the medium cost is brought out to as low as 8 rupees or 10 rupees maybe. So you can see the vast difference. So this also by which we can reduce the production cost. And lighting, this may not be feasible in a tropical country like ours, but like because you have so much of solar radiation, which is increases the temperature and which is not favorable for the culture development. But if you see in countries like Cuba and Austria, where they go for solar illuminated tissue culture rooms. They don't use any tube lights or the fluorescent lights or LED bulbs, nothing. They just use give solar illumination for the tissue culture rooms, and greenhouse with additional shade net. This is also possible. You can see this is just a glass house with a shade net here, or a black shade net. You can see. So what they do is, whenever the light intensity or when the sunlight is more, they try to close the shade net, and whenever the light level is low, they try to open up and give more light. So this is how, and this they use it only for the rooting phase, not for all phases, only for the rooting phase. And rooting and primary hardening. So as I said earlier, you have to take care that you can do in the compressed coco pit, you can do both primary and secondary hardening at one go. And you should also take care of the irrigation, whatever is being used for hardening, because most of the times they also carry nematodes, so which is likely to affect the tissue culture plants. So you have to take care that you give good water for irrigation. And all these points I have already told you. And this is another technique, we call it as a double layer technique. This is again to minimize the labor and the time, because what we do is, once single plantlets are developed, we need to, as I told, we have to go for an exclusive rooting medium. So taking out these plants, separating them individually and then putting in a rooting media, it is time consuming. So normally what we try to do is, we take them, once the single plantlets are formed, we take them to the laminar airflow chamber, we just pour the rooting media in the same media. So it will be in the liquid form. So that also by which you can save labor as well as the energy. So normally the tissue culture plants, we need to grade it, and accordingly we need to put them in the pro tray, so that there is not much ununiformity in the age of the planting material and the growth. So all these points you have to take care. And these are all already I have discussed. And what should be an ideal tissue culture plant? Because normally we say it should have 5 to 6 fully open leaves, and the collar girth or the stem girth, and it should be at least a minimum of 5 to 7.5 centimeter girth. And we see that it has 25 to 30 active roots, and we also should ensure that they are free from root knot nematodes. As I told, that the irrigation water might also carry nematodes. So all these points you have to take care when you select an ideal tissue culture plant from the nursery. So then as such, you have to look for the somoclonal variants, and also you have to look for the freeness from pest and diseases, visual freeness. And this is a secondary hardening one. And this is a recent study what we have made with respect to banana, because this has been studied in other crops, whether we can replace the white fluorescent lamps which we are currently using with the LEDs. Yeah, definitely we can use it, because many companies are using, but still we should prove them scientifically before we try to advocate to the tissue culture companies. So you all know that this LEDs, they have a superior light quality, and the energy consumption is very much minimum, so it can ultimately reduce the production cost, and the heat generated is also low when compared to the heat generated while using the white fluorescent lamps, and lifetime of the LED bulbs is also more. So what we have done is, we have tried all the different explants in different different colored LEDs, that is different spectral wavelengths we have tried, and we found that for each of the explant and for the stage of the development, the light requirement varies. That is what I am showing it here. So if you take the bud proliferation was better. This is what I was talking about, the direct regeneration using the male flappets. It was better in case of red LEDs, and the bud proliferation was better in case of green LEDs also, followed by red color. And here also you can see the meristem clump. meristem clump formation was better in red LEDs followed by green LEDs. So even the somatic embryos, that also the germination was influenced by the spectral wavelength. And now coming to the somatic embryogenesis. You know, they are the embryos which are formed from the somatic cells other than the reproductive cells. And the structure of the somatic embryos will be very similar to that of the zygotic embryo, and they will go through all the stages like globular, heart-shaped, and the torpedo stages. Again, these are also totipotent in the sense from which you will be able to regenerate a whole plantlet. And zygotic, normally we say that zygotic embryos, the plants are not genetically identical, but here the plants which are derived from the somatic embryos, they are genetically identical, and which we have proved by conducting the field evaluation studies. And this is again can be used as a high throughput method for clonal propagation or for tissue culture multiplication. And as I told in my very first slide, this is the basic in vitro system which is required for genetic transformation and mutation studies. And what are the stages? So you can see the first stage, this is from again from the male flower bud, which I'll show in the very next slide. So the first phase is the callus induction, and next phase will be the establishment of the cell suspension in the liquid culture, and third will be the regeneration, we call it as, or the maturation of the somatic embryos, because here they will be just immature embryos. Here we need to mature them. Then final comes the germination phase. So as I told earlier, that's what I was telling you, that I'll be showing a slide where you will be able to see the size of the flower bud which is being used for the in vitro culture. So after removal of all the outer bracts, we can bring down the size to 5 centimeter length. Then you can take them to the laminar, swab with the alcohol, and then you can try to pull out the floral hands. So, but you have to use appropriate media. So the media composition only determines whether it has to undergo direct regeneration or it has to undergo indirect regeneration, where it is rooted through callus. So callus induction will happen, and from which you have to develop the embryogenic cell suspension. So factors that influence genotype naturally, because as far as banana is concerned, it is a highly complex crop where everything depends on the variety and the genome, and as well as the genotype. And explant what we use, because different explants can be used for somatic embryogenesis. You can use a scalp, and you can use leaf material, but they are all not commonly used, but they have tried. And what is commonly used is the male flower bud, immature male flower bud. And media composition, as I told, for different stages of development, it needs to have a unique media which is supplemented with appropriate growth regulators. So that is what is the thing. Then temperature and the light intensity. Temperature, 26 plus or minus is always good for all in vitro culture studies. And light intensity, normally it is we say 2600 to 2000 lux. But what happens is, as far as somatic embryogenesis is concerned, and whenever we go for the indirect regeneration method, that is when we try to develop the plants via callus, and then we go for somatic embryos, normally the callus induction phase, all phases they are done only in dark, except the germination phase, which is the final phase, which would be carried out under light. So that is what is important with respect to somatic embryogenesis. And you can see, like if you see granine, it has a callus induction was maximum, that is 13.3, and followed by Rastali, Nendran, and then Udaimet is a Karpuravali type. So the embryogenic callus is more important, because that only gives rise to the cell suspension, not the non-embryogenic callus, which will be just watery, and they will not be able to regenerate into a plantlet. And I was talking about the different explants. Even the meristem can be used to develop the scalp, we call it as, that is, it will be a proliferating meristem we can make, and that will be a can be used for the induction of callus and leaf explants. But the common one which is used is the floral hands. So if you see the difference, you can see that 1 out of the 3 good male flower, it resulted in callus development, whereas in case of scalp, 1 out of 5 good scalp only, it resulted in the callus production. And media composition, that is more important, especially your growth regulator, that decides everything. And this also I talked about, where you should need to have 16:8 light bar dark cycle with the 2000 lux intensity, and 26 plus or minus degrees and 2 degrees centigrade is more important. So I was talking, that as 8 to 16, even not 8 to 16, it is, you can keep it as 10 to 16. From the outside world, you have to count. When the male flower buds is 5 centimeter, you have to start counting from the outer world. If you go from the outer up to up to 8th world, from 8th to 16th world, it shows a good response for the embryogenic callus production. So when you use the appropriate medium for the callus inductions, what happens is all the from 1 to 7 world, it undergoes necrosis, while hands from 17 to 20, again it turns brown because of the high phenol secretion. So they cannot be used for the production of the embryogenic callus. And the required hormones normally IAA, NAE, 2,4-D, and even zeatin also we use. And how do we identify a good callus for establishing the cell suspension? So when you put them floral hands, you will get the callus. So this callus development, it almost takes 3 to 5 months depending upon the variety of the genotype. So if you see, these are all the embryogenic calli which are very translucent, and beneath this is, you can see the pro-embryogenic cell mass, we call it as. So you have to abrase this, and then you have to take out these cells and put them in the liquid media so that you develop a good suspension. And in case of the you can see 2 types of cells. That is, one is the cells, the prominent cells with the nucleus and nucleolus, and other one you will have more starch granules. So you can check the viability. So when we use the DAPI strain, all the nucleus, it turns blue in color. And when you go for the starch granules, cells with good amount of starch granules, when you strain them with Lugol's iodine, and the cells starch granules will turn into deep purple in color. So then we can confirm that these are all the embryogenic calli, and that we can use it for the development of the suspension. So these are the other non-embryogenic calli which are produced, and these have to be avoided. And if at all you initiate them, it is going to be of no use, and you will not be able to end up in embryogenic callus or plants. So as I told earlier, this is again, it is the embryogenic callus and the pro-embryogenic mass. We put it in the liquid media, and then we develop the suspension. So like NRCB, it has developed the protocol, because everything is genotype dependent. So we have to have a unique protocol for each of the genotypes, whatever you are interested in multiplying. So it has developed the protocol for almost all the commercial varieties, and we do the multiplication. And initial stages, because until you are able to establish a suspension and it starts proliferation, it is good, highly laborious in the sense. Initial stages, you have to subculture them in alternate days, and then you have to remove all the dead cells, brown cells, black cells, everything we need to remove, so that you end up only with the fine cells which are good and good regeneration potential. And once it starts proliferation, then subculturing can be done once in 7 to 10 days. And we need to check the viability, because we can maintain these embryogenic cell suspension up to 2 years. But just before initiating, you have to check that it is quite viable. By doing DAPI staining you can do, or you can go for fluorescent diacetate staining, where the viable cells, they fluoresce under fluorescent microscope. Then optimum conditions, because normally it is kept in a shaker, in the sense which with 70 to 110 rotations per minute, so that the cells are not damaged, and they multiply, and we developed a fine homogeneous cell suspension. And this is what I was talking about, the fluorescent diacetate staining, and how it slowly develops. This is after a week, and how it develops into granular type, that is maturation happens. Then this will go for germination. So this is the germination you can see after 10, 30 and 45 days, and a whole plantlet developed after 45 days. So just if you see here almost 30 days and here another 45 days, almost for the maturation and germination alone, it takes 75 to 90 days. So the that can be reduced if you are able to use for both the maturation as well as the germination, if you are able to use a liquid media, because here they will be in direct contact with the media and there will be effective absorption of nutrients from the media, and you can enhance the germination percentage. So, and the plants produced were also comparatively bigger, and the problem of what you call it as vitrification problem was not observed. And risk of when we use a liquid culture, the risk of contamination is high, and we need to subculture once in a week. That is, again, it is quite laborious, but still, if you want to develop earlier planters, then you can go for the use of liquid culture for maturation as well as germination. And now this is a advanced system, that is where we use this we can use this temporary immersion system bioreactor both for the shoot tip culture as well as for the maturation and regeneration of the somatic embryos. So here normal like what we do is periodic immersion of the explants in the nutrient medium, which results in uniform distribution or diffusion of nutrients, and gaseous exchange will be improved. So naturally what we try to do is we do that immersion in the media at programmed interval, and the time for which it has to stay in the media, that also is matters, and it synchronizes the growth rate of the plants. So here, as such, NRCB, we have developed two types of bioreactors. This is not a temporary immersion type. This is a bubble column bioreactor where 10 ml of the cell suspension can be multiplied 11-fold. That is almost 110 ml within a short span of 21 days. So that is the advantage. So otherwise, in a normal case, if you want to multiply, when I showed you the easiest or all the cell suspension in conical flask. So if you try to multiply that way, then to achieve this 110 ml, it almost takes 2 years period. But when you are using this bubble column bioreactor, the greatest advantage is within 21 days, you are able to multiply 11-fold. And this is we have developed a low-cost, customized, temporary immersion type bioreactor, which will maybe in the next picture you will see that you'll be able to see the whole structure of the bioreactor. And here you can use both the shoot tips as well as the somatic embryo maturation and germination can be carried out. So what is the advantage? So repeated subculturing is not required, because then when repeated subculturing is not required, naturally the manpower and the production cost will be reduced, and scaling up of production is also easier, and you'll be able to produce a quality material, planting material within a short span of time. So this is a customized bioreactor what we have developed, and we have even filed a patent, hoping that we will get the patent shortly. And this will have the nutrients in the lower flask, and this will have a microfilter here, so that when it is put in the culture room, there is no entry of pathogens into the media or to contaminate the plants, whatever is being developed. So all these will be connected to a motor. That is, it will be a small motor, 0.5 HP motor, a series of 5 immersion systems, or the bioreactors could be connected to a single motor, so that what happens is it pumps the liquid from the lowermost flask to the upper flask, and here there will be a filter, so which I filter, so that the plants or the embryos, because I told even maturation can be done. This is the germination stage. So there are no chances for the plants to get back into the nutrient media. But when they are in the cell, that is maturation phase, there are every chances for the cells to get back into the media. So that may not happen because of the provision of the filter here. So what is this again? Because here you like from 1 ml of the suspension, this is not even 1 ml, it is actually 0.5 ml. You are able to produce almost 2000 plants, and the problem is because of the congestion, all when you grade them, what we get is only 50% of good quality plants, and rest of them will be very small. So to avoid that, we wanted to provide a adequate space or the volume for them to grow luxuriously, so that all the plants, all the 2000 plants which are produced here are of uniform length. So this is the potential of this particular bioreactor. So these are all the commercial bioreactors which are available and which are being used for banana. This is the Rita type which has been developed by CIRAD people from France, and this is another type of Plantima bio. Shall I go ahead, sir? So as I said earlier, this bananas like are highly susceptible to somoclonal variation, because most of the somoclonal variants, they are negative in nature, in the sense most of them they didn't come to flowering at all, or they produced like low quality bunches which cannot be marketed. So all these are the somoclonal variants, but there are every chance that we can positive somoclonal variants we can make use for the improvement programs, in the sense like if you take Taiwan, they have isolated this. You would have heard about the Jane Cavendish tissue culture variant, GCTCV, they call it, as different lines they have which are resistant to, likely to be resistant to fusarium wilt. So all of them, they are positive somoclonal variants, what they have developed, and they have tested for on the wilt. So that is also possible, but most of the times it is negative. It again, the extent of variation, it depends on the variety and the number of subcultures, because when you go beyond 8 subcultures, you will be able to produce more plants, but according proportionately, the number of or the percent of somoclonal variants occurrence also will be high, and subculture interval also it matters, and growth regulator concentration, because normally we recommend we should never go beyond 5 milligram BAP for at any time of the tissue culture plant development. But there are companies who use more than 5 ppm, that is 5 milligram per liter. So then naturally what happens is the percent occurrence of the somoclonal variation is more. So in all such cases, we have to take care, and we have to avoid such negative off types, and we have to minimize the somoclonal variation, because if it happens, then it becomes a problem to the farmer who grows the tissue culture plants. And initial days, like they were using like spuriously this growth regulators and the number of subcultures what they make. So it all resulted in higher percentage of somoclonal variation, even up to 60%, where the farmers were at a loss, and there were lots of hues and cries, and they even sued the court for getting compensation. So, but now after the intervention of the DBT, they have made a certification as important. As I told, we need to index them for viruses at different stages, and we have to do the clonal fidelity at the ICAR plant. By doing all these and streamlining all these processes, they were. I can say that they were not able to totally bring down the percent occurrence of somoclonal variation to 0, but still they were able to reduce it to a greater extent. So now still there are problems in certain pockets, but still it is under manageable limit. So these are all the possible off types that could be seen in the nursery as well as in the main crop. So dwarf and short statured plants. And normally granite, it is supposed to be a Jane Cavendish type, but if they look short and dwarf, then we have to cull out. And unusually long peduncle. Maybe these I have listed. I'll show some of the photographs. So short stature plant you can see here. This is a short stature plant when compared to the next plant, which is quite normal. And you can see the rubbery nature or the variegated leaves you can see, and some sort of mottling. And you shouldn't get confused for the micronutrient deficiency as well as for the variation. You have to be very clear. And deformed leaf blades you can see. And you can see the split and crooked fingers. All these you have to avoid. When you take the explants also, you have to take care of all these points. And these are a few of the somoclonal variants. And this is about the embryo culture, because why this is, I have mentioned about the wild species. Wild species as such, if they germinate under natural environment or the natural habitat, they germinate. There is absolutely no problem. But when we try to bring those seeds and try to germinate them here under sand, it is not happening, mainly because of the missing of some of the soil microflora and other aspects. So you can see the seed size. It is ranging from very like small seeds to bigger ones. And what you need to do is, we normally say we need to prime the seeds. That is, you have to soak them in a liquid water, or you can go even for hormonal priming. And we have found out that GA3 was giving better results. When you prime them with GA3 at 15 ppm, then you grow it under in vitro condition, the percent germination could be enhanced. Because normally, like when you put them under the sand bed or the in vivo condition, what happens is the percent germination is very, very minimum. It is less than 1%. So you have to, if you want to enhance to higher percentages, then definitely you have to go for embryo culture. So this is how the embryo is embedded in the seed. Then this we have developed into a plantlet by which we are able to enhance the germination percentage from 7 to 10%. So this is how we have developed the plantlet. This is more important, especially when we develop the progenies, because when we do crossing and get the seeds after almost a span of 1 to 1.5 years, and if you are not able to germinate, then the whole process goes waste. So in such cases, you can go for the embryo culture. And also we have developed protocol for developing multiple shoots from the single embryo. So that is also possible, so that at one point of time you can produce more number of plants from a single event, and all of them can go for evaluation by for different biotic and abiotic stresses. And this is with respect to production of haploids in case of banana. So we have tried anther culture, and what happened was, unfortunately, all of them resulted in the production of tetraploids. This might be because that we have extracted the anther when they were in tetrad stage. So that could be one of the reasons. So you have to go for uninucleate stage. That is the most important. But unfortunately, we have taken a tetrad stage, which resulted in the production of the tetraploids. But here also, like, we could make use of them, because tetraploid, when it became tetraploid, all of these were their fertility was enhanced, male fertility exclusively. So we can use them in the crossing, so as to produce the commercial triploids. So I was talking about all these explants from where we can produce the plants from comlet, male flower bed, and shoot tips and suckers. So there was not much difference in terms of yield. All of them, they were uniform. Next one is, so what I conclude is, by adoption of quality planting material, you will be able to enhance the production and productivity, so that the farmers get good returns. So these are all some of the varieties which has been released by NRCB. And thank you very much for your patient hearing. I think I have taken exactly 1 hour.
Yes, madam. Thank you so much, madam.
Yeah.
Now I request Dr. Vinay Kumar Reddy to please propose formal vote of thanks.
Thank you, madam, for giving the opportunity. So I thank Saraswati, madam, for giving a detailed lecture on tissue culture for production of the quality planting material for the farmers. And Madam has explained in detail about the tissue culture, how to select the explant, and also what are the media to be used and the cost involved in the media, and also how to propagate it and how to harden it, and also various techniques which are involved in the tissue culture, like bioreactors, and also the somatic embryogenesis, and also Madam has explained in detail all those things. Thank you, Madam. Thank you so much for giving a detailed lecture and also enlightening our students with advances in tissue culture technology. And I also thank Dr. Ramanandam sir, Principal Scientist, HRS Cover, and also the participants, the student participants. So with this, I thank once again Saraswati Madam for giving a beautiful presentation with good pictures to explain in detail about the tissue culture to our students.
Sir,
Thank you, madam.
Thank you very much, sir. I also thank the organizers for giving me an opportunity to be a part of this graduate readiness program, sir. And I wish that at least you people, they should get benefited by this lecture, and they should venture into the tissue culture companies, and they should become the entrepreneurs of future, because the demand for quality planting material is increasing day by day, not only in Andhra, but also in Tamil Nadu and all parts of the country. Because though they are the plant cost is a bit higher when compared to the conventional suckers. Now, if we say almost it is equal, sucker cost as well as the tissue culture planting material cost. So they have to make use of this, and I would like to see few entrepreneurs, budding entrepreneurs, by hearing to the lecture, and maybe for some it may be inspirational. I don't know, but anyhow, I also thank the organizers for the opportunity, nice opportunity given to me to share my experiences with the student community. Because normally we talk only to the scientific community and other groups, but to the student community, it is. I'm very much happy, sir. Thank you very much, sir.
Thank you, madam. Thank you so much.
Madam, shall I end the session?
Madam, thank you very much, madam, for your very.
Welcome, sir.
Yes, madam. Thank you very much, madam, because this is our Honorable Vice Chancellor's brainchild program, madam. Actually, this year, Sir has declared Year of Banana,
Yeah,
and
yeah.
he has started this one. He is instrumental in conducting this program.